Hello, my name is Gabriela Schlau-Cohen. I'm an associate professor in the Department of Chemistry. The stories I'm going to focus on today all involve using single molecule spectroscopy as a tool to investigate the conformational dynamics of membrane proteins. What I'm showing here are the top 10 best-selling drugs of 2017. And if we look at this list, what we can see is that 7 of them are membrane proteins. In fact, over 60% of drug targets are membrane proteins. But that comes with a challenge. If we think about building a mechanistic picture of how these membrane proteins work, we can look at their structure and see that the extracellular and intracellular regions are hydrophilic. But that's in contrast to the transmembrane region, which is hydrophobic. So this design, this mixture of hydrophilic and hydrophobic regions is challenging because it requires maintaining that complexity, those two very different environments, while conducting experiments. And so for mechanistic experiments, which often require in vitro environments, that mixture isn't available. And if we think about studying membrane proteins, one, their fundamental function is to detect the chemical state outside the cell and then transmit that information into the conformational or reactive state inside the cell. But because of the challenges involved with membrane proteins, what's often been done is these structures are taken and then chopped up into individual domains. Now, these kinds of piecewise studies of individual segments have been insightful. There have been a lot of new information about the different conformations of the regions themselves. But in these kinds of piecewise studies, this fundamental information of how the extracellular and intracellular environment is connected is fundamentally inaccessible. And so that's why one of the things that we do in my group is build structures like the one you see here, where we take this membrane protein, we embed it into a lipid bilayer, and then we solubilize that entire membrane and protein structure with a belt. In this case, the belt is an apolipoprotein. So this is an emerging biochemical technology called a nanodisc. And nanodiscs are self-assembled, so there's control over the size of the disc, the composition, including the lipids or the addition of cholesterol. And we can control the proteins inserted. So these provide a near physiological environment to look at membrane proteins, where we can have both the hydrophobic and hydrophilic regions, but we're able to perform mechanistic experiments in vitro and without the background of many extraneous cellular processes. So we have a well-controlled sample to study. And within this well-controlled sample, we have access to both the extra and intracellular domains, which allows us to investigate those fundamental questions of connectivity. So what we do in my group is we take these constructs and we attach fluorescent dyes to different points of interest on the protein or on the membrane bilayer. And then we can use a tool called single molecule first or resonance energy transfer, or single molecule FRET. This provides a nanoscale ruler. We can measure the distance between two fluorescent dyes using their emissive properties with a properly calibrated sample. So that distance gives us a reporter as to the protein conformation. We can monitor it and look at the conformational dynamics. And we do this at the single protein level, because that allows us to identify the conformation of an individual protein and its dynamics that are often averaged out in a heterogeneous ensemble. In an ensemble measurement, you may have 10 to the fourth individual proteins, each at a different stage in the signaling pathway. And that averaging prevents you from monitoring the individual steps and their sequence throughout the signaling process. So getting back to this picture of the top 10 best-selling drugs, I want to begin by focusing on this one, Herceptin. Herceptin, as many of you know, is part of a class of drugs that have emerged as an exciting alternative to traditional cancer treatments. And these molecular or targeted therapeutics often target members of the EGFR, the epidermal growth factor receptor family of proteins. So these receptors regulate a host of cellular processes in mammalian cells, including growth and proliferation. And they've been implicated in a range of cancers. Lung cancer, over 60% of breast cancers. And so it's because of their role in cancers that they've become the target of Herceptin and others in its class. But if we want to think about how EGFR works, one thing that's been emerging recently is that it exists in multiple oligomerization states. Now, EGFR is well known to form a dimer structure. Ligand binding leads to dimerization, and EGFR can signal within its dimer structure. But there's also multimers of EGFR that form and are increasingly thought to have a functional role. And EGFR can also be in its monomeric form. So it's well known that there's a heterogeneous cellular environment that may play a role in these different oligomerization states. So if we're thinking about EGFR function, we can begin by thinking about the monomer as the simplest transmembrane structure for us to investigate mechanistically. So here's a structural model of EGFR. And so the question that we want to understand is, what are the conformational dynamics in the monomer alone? So this is part of the series of experiments that we do in my lab to build up a mechanistic understanding of the function of these membrane proteins. So it's been well established in EGFR that there's a series of conformational changes in the extracellular region. For example, upon binding of the predominant ligand, epidermal growth factor, or EGF, there's an expansion of the extracellular domain. But what specific conformational changes, if any, in the intracellular domain have been much less accessible? And so we address this question through a series of experiments. I'm going to tell you about a couple today. One of them looks at the conformations associated with the binding of ATP. The intracellular domain, there's a kinase region that binds ATP to drive signaling. And so using single molecule FRET, we looked at the distance between these two fluorophores as a reporter of the intracellular conformation. And so here's the histogram of conformations that we measured from individual proteins. So this was constructed by measuring the distance between the ATP binding site and the C-terminal tail, where we attach a different fluorophore for one individual protein. Then we did it again for another, and then another, and then another. And that allowed us to build up a distribution of this distance here, which captures the overall conformation of the intracellular domain. And so what's shown here, as I said, is that histogram. What we can see is it's relatively wide. This overall conformation, this distance, ranges from 7 to 11 nanometers. So this wide range of distances likely reflects the heterogeneity associated with multiple phosphorylation sites and also with a disordered tail. So this C-terminal tail is an intrinsically disordered region of the protein structure, which introduces additional heterogeneity because it's lack of well-defined structure. So this C-terminal tail, shown here, is a disordered region, but it also contains the tyrosines that undergo phosphorylation in a ligand-dependent manner, and it's those phosphorylated tyrosines that dock signaling proteins to activate those signaling pathways. Now, the C-terminal tail has this disordered nature that's thought to play a functional role because disordered structures are thought to be able to interact with much more diverse signaling partners. And EGFR, like I said, is known to regulate a host of cellular pathways, so that conformational flexibility associated with its diverse disordered structure may enable that flexibility in interactions with signaling partners. However, although it is functional, that disordered nature means that structural characterization is challenging. There are very few ensemble structural methods that are able to characterize intrinsically disordered regions. The only thing that's known because of the limitations of those biophysical methods is that there's a local expansion associated with loss of a transient secondary structure that was identified upon phosphorylation. But this region is predominantly disordered, so that transient secondary structure does not well describe its overall conformational state. So we used our technique of single molecule FRET to investigate its structure and structural dynamics, where we created a construct of the C-terminal tail alone, illustrated here, and we attached fluorescent dyes. We repeated our single molecule experiment for the cases where the construct is dephosphorylated and chemically phosphorylated. And here are the histograms, where we created 2 different samples and then measured many different individual constructs for both the dephosphorylated and chemically phosphorylated constructs. And in these distributions, what we saw is that they are overall wide. And this isn't surprising because this is likely due to the structural heterogeneity associated with this intrinsically disordered region. This is furthermore likely due to the same heterogeneity that gave rise to the wide distribution that we saw in the case of the ATP to C-terminal tail distance. But along with it, what we saw is that upon phosphorylation, the average distance went from 10 nanometers to around 8 nanometers. So this construct had an overall compaction of around 2 nanometers upon phosphorylation. And so this observation was in contrast to previous models where a local expansion was observed. And it was assumed that that reflected an overall expansion that basically opened up the C-terminal tail to enable the docking of the signaling proteins. So to investigate what happened, we turned to coarse-grained MD. Here's 2 snapshots from those structures. These are actually snapshots selected from the most common overall structural motif. And what these show is in the dephosphorylated case, you have a disordered region lacking any structure. But in the phosphorylated case, the negative charge of the phosphorylated tyrosine interacts with 2 neighboring positively charged residues, shown in blue. The tyrosine is shown in orange, causing the formation of a conformational loop. And so what's notable here is that in going from dephosphorylated to phosphorylated, you have a conformational change where you form this loop driven entirely by electrostatic interactions within the C-terminal tail. So thinking about the overall intracellular conformations of EGFR, what we see is that there's an overall heterogeneous conformation due to the disordered nature of the C-terminal tail. And we see a phosphorylation-dependent conformational change within the C-terminal tail. Now that conformational change, the formation of the loop, may be something that activates the docking of signaling proteins. But it creates an architectural motif that may facilitate the docking of signaling proteins while retaining the lack of secondary structure or the overall disordered nature, which is a prerequisite for the ability to interact with diverse signaling partners. So this may be how EGFR is able to activate signaling, but still activate signaling with many different binding partners to regulate the multiple different cellular processes for which it is responsible. So overall, collectively, these experiments have given us a picture of the different intracellular conformational states of EGFR. Now I'm going to switch gears and tell you about a different story where we use similar techniques to look at a different problem of signaling in bacterial cells. So it's been well established that antibiotic resistance is a growing crisis. There are 2.8 million bacterial infections per year in the US, and it is incredibly widespread, so widespread it's not even well characterized globally. Now, if we think about bacterial infection, one thing that we know about bacteria is that many are motile. Those motile bacteria swim towards favorable conditions, such as food, and which is what begins infection. So interruption of the signaling that controls the swimming could be a mechanism to block that first step of infection. So how this motion called chemotaxis works is in the absence of attractants, motile bacteria undergo this tumbling motion. In the presence of attractants, bacteria move along chemical gradients in a straight swimming manner based on the direction of rotation of their motor. And so what we did is to look at the first step in this motion process. We looked at the chemoreceptors. This is the receptor protein that binds attractants or repellents. We looked at the receptor for aspartate, a sugar, and that's shown here. So the fundamental function of this receptor, again, similar to EGFR, is to take extracellular ligand binding and then transmit that through a series of structural changes into control over an intracellular signaling pathway. And so we looked at the chemoreceptor dimer, which is the simplest active structure that can bind ligand and then transmit that into intracellular information within the chemoreceptor. And so cytoplasmically, it forms this 4-helix coiled coil region. And so within this 4-helix coiled coil, there's N helices shown in orange and then the C helices shown in blue. And we created 2 different samples, one of which we attached fluorophores to the N helices and the other of which we attached fluorophores to the C helices. And so we were interested in looking at what is the cytoplasmic change upon periplasmic binding of aspartate, the sugar. And so to begin with, we characterized the distance between the 2 helices in the absence of aspartate. And what we saw is that the C helices, shown on the right, are closer together with an average distance of around 50 angstroms. The N helices are further apart with an average distance of around 60 angstroms. This tells us that this 4-helix coiled coil region forms a rhomboid structure. In the presence of aspartate, what we saw is that effect increased, where the C helices moved even closer together to around 45 angstroms, whereas the N helices moved slightly further apart to around 65 angstroms. So that rhomboid structure increased with aspartate. This tells us there's a change in helical packing. We can also look at the dynamics. These are the conformational fluctuations in each signaling state, which reflect on the helical dynamics or helical flexibility. We saw, in agreement with previous work, that the N helices were more dynamic. And then upon the shown as a faster timescale of switching between conformations, then in the presence of aspartate, we saw that that effect increased further, that the N helices got even more dynamic, where they switch between conformational state on a even more frequently, whereas the C helices exhibited similar switching dynamics. So collectively, what these changes tell us is that there's concerted changes in the helical packing and dynamics, where the packing goes from a modestly rhomboid to a more extensively rhomboid structure, and the more flexible or more dynamic set of helices becomes even more dynamic, whereas the other helices presumably serve as a stable scaffold pair. So both of these sets of changes in packing and dynamics are consistent with this overall conformational change from a slightly rhomboid to a more extensively rhomboid structure. That more extensively rhomboid structure may free the N helices to enable them to open up and interact with signaling partners. So collectively, what these 2 sets of experiments tell us is that by using these membrane discs called nanodiscs, we can construct well-defined systems that will allow us to map the spatiotemporal dynamics of signaling through membrane proteins. This historically challenging class of proteins to study. And by using single molecule FRET, we can monitor their conformations and characterize their conformational dynamics. We've been able to use this approach to determine previously inaccessible intracellular conformational changes in both bacterial and mammalian signaling. And these previously hidden motions may be playing a functional role in allowing interaction with signaling proteins um, to control cellular processes. So finally, I'd like to acknowledge the people who actually did the work. Here is a Zoom photo of the group. The first story I told you, the work on EGFR, was begun by a really talented former postdoc, Steve Quinn, who's now a lecturer at the University of York. That work has been continued by a current postdoc, Raju Regmi, as well as a graduate student, Shweta Srinivasan. That work is in collaboration with Matt Coleman at UC Davis Med School and Ron Bose at Washington University School of Medicine. And the MD simulations were performed by Bin Zhang, my colleague at MIT. The second half of the work on chemotaxis, was originally led by a former postdoc, Julian Troiano, who's now at Intel and has been taken over, and really the new insights have come from the work of talented graduate student team, Jesse Gordon and Michaela Hoffman. And I will say that all of these single molecule experiments were enabled by foundational work in my lab, building up our ability to do single molecule experiments, by a phenomenal former postdoc, Toru Kondo, who's just taken a position as associate professor at Tokyo Institute of Technology. And finally, the work on chemotaxis is in close collaboration with Gerald Hazelbauer at the University of Missouri. These are the current funding sources for my lab, and I am happy to answer your questions.